my presentation on MSProGene as a new method for integrative proteogenomics. I will start with a very brief introduction of the field of proteogenomics. So what do we mean when we talk about that? I think that all of you, or most of you, are familiar with the field of genomics, where we look at genomic sequences and we try to annotate structures, for example, genes or their regulating mechanisms on these genome sequences. And following the dogma of gene expression, these genes are transcribed to transcript sequences, and we can use NGS technology, for example, the RNA-seq, which gives us millions of short reads to reconstruct these transcript sequences. And this is like summarized under the field of transcriptomics. And again, following the dogma of gene expression, these transcripts are translated to protein sequences. So we look in the field of proteomics, we look at these amino acid sequences. And for example, we can use mass spectrometry and tandem mass spectra to first reconstruct the peptide sequences and then infer the original proteins. And now, if we combine several of these omics data, nowadays even, for example, metabolomics or interactomics can be used, then we talk about the proteogenomics field. And this is what we are also targeting with MSProGene. So we try to combine not only genomic information and proteomic information, but also using transcriptomics for an improved genome annotation and protein identification. And because there's such possibility in this combination in using different data, there's a wide variety of studies that evolved over the last years using proteogenomic measurements. These are just few examples of all the available studies and also some pipelines that evolved over the last years. And as you can see, there's a great need for sophisticated methods that integrate these different kinds of data. So what is common to existing methods or existing studies is that usually for the protein identifications, for the search of MS2 spectra, they use existing reference databases of protein sequences. And normally you use known single nucleotide polymorphisms, for example, annotated in databases like dbSNP, to extend these existing reference databases. And what is also quite common is to include all 6 frames for the translation as depicted in the figure on the right-hand side. What is also possible with increasing availability of RNA-seq experiment is to use the expression information or the information of SNPs that arise during this RNA-seq experiment or that were observed during the experiment, and also splicing information to also extend the databases. What is the problem here is that these databases might be incomplete, so we might not be able to actually identify novel proteins that are not known before. And of course, known annotations are not very suitable for an experiment-specific analysis. So for example, to compare different conditions of a specific cell and the response to that. And what is also a problem is that 6-frame translations can introduce a bias to the peptide identification. And another aspect is that the efforts today are mostly focused on an improvement on the peptide identification side, but also the inference of proteins is quite a problematic thing. And there are several difficulties, for example, shared peptides that lead mostly to a protein grouping approach, which might also be, or here it might also be very good to find better ways or more sophisticated ways to improve the overall identification. So MSProGene was designed to try to overcome some of the current problems that exist in these proteogenomic studies. And what we do is we take as input a reference genome and an experiment that yields RNA-seq reads and corresponding MS2 spectra. And then we first construct a customized transcript database based on this specific experiment, so using the reference genome and the RNA-seq reads. And then we perform a search of the MS2 spectra against this customized database. And then we enter a processing phase where we first analyze the peptide identification results and then perform a quality control. To be a bit more specific, so first, as for the reconstruction of the transcript database, we use, for example, existing methods like JIRA or the well-known cufflinks, which result in nucleotide sequences that can be analyzed. And we include SNPs and insertions and deletions that we observed in our RNA-seq mapping to these reconstructed sequences. And we perform, which will be important later on, we perform an initial scoring based on the observed read coverage of these transcripts. And then for the following spectral search, we need these nucleotide sequences to be an amino acid format, so we perform a 3-frame translation, so we can avoid a 6-frame translation as much as possible, which is only required if we have no information on the specific strand of the transcript. So then we use existing methods, for example, the MSGF+ method for the peptide identification on these transcript sequences. And here you see a very, very simple example of a possible outcome. So we have 3 peptides, P1, P2, and P3, which map to 2 different transcripts. And here I want to point you to the 3 main objectives that we have to tackle when we want to analyze these peptide identification results, which is first, as you can see here with peptide 1 maps to 2 different transcripts. So we have to identify the correct transcript for this peptide. And these peptides are also known as shared peptides. So we have to identify the correct origin. And then we have the case, for example, here for peptide 2 and peptide 3, they map to 1 specific transcript, but to 2 different frames. So we know beforehand that one of these 2 identifications must be incorrect, because it's not possible that we have 2 frames for 1 transcript. So the second thing is to identify and correct for the incorrect peptide identifications. And of course, we also want to identify the correct frame for each transcript that we reconstructed beforehand. And to do so, we reconstruct a proteogenomic network based on the peptide identification, where we first introduce a node layer representing the peptides. Then we have nodes representing frames, and peptides are connected to frames by a directed edge whenever the peptide was identified for a specific frame. And what is also important, we label each connecting edge with a term that specifies the capacity of this edge. And the capacity is a throughput that is allowed to be passed from one node to the other. We also introduce a transcript layer, also with an associated capacity. And then, for technical reasons, we introduce an artificial node called the dummy node, which serves as a bin for the incorrect identifications. So each and every peptide is connected to the dummy node, which is an artificial connection that is only chosen in case our algorithm that will soon proceed through this network identifies a connection as incorrect. So this is only to filter out those incorrect identifications. And then we want to perform a maximum flow on this network. So we introduce a source node S and a target node T, and then we pipe flow starting from the source towards the target node through our network. And the maximum flow is interested, of course, that this name is interested in maximizing the throughput that can be passed through the network. And we chose a formulation as an integer linear program because in the end, this will result in the edges or in a choice of edges that maximizes the throughput. And this is corresponding, hopefully, to the most likely connection for each peptide. And I just want to briefly point to some important aspects here. So I formerly mentioned the transcript coverage and the initial scoring. So this can be found in the network. So initially, the edges have a certain capacity that is based on this transcript coverage. And we also have of course, an initial scoring of our peptide identification, which also can be found labeling the edges connecting peptides to frames. And the only exception of this case is the dummy connection, where here you can see that outgoing of the dummy, there's an unlimited capacity. So in principle, everything could be piped to the dummy. But this would not be wise because we introduced a penalty that says, okay, we will penalize the overall flow whenever we choose such a connection. After that, we identified certain connections as the most likely ones. So for each peptide, we have a new association to the most likely frame. And now in proteomics, this is quite, or you might be quite familiar with that, we need to perform some sort of quality control on our remaining set of peptides. And usually you use the false discovery rate derived by a target decoy approach to do so. But in our case, we have no decoy sequences available anymore because we were only looking at target peptide identifications. So we have to find another way for the quality control. And we do so by assuming a mixture of targets, so true identifications, and decoy identifications, so false identifications. So we assume our set of all scores, which are depicted here in the histogram, to be a mixture of these two distributions representing true and false identifications. And we want to fit these two distributions, in our case, using an expectation maximization algorithms to the complete set of scores. And by doing so, this allows us to calculate a cumulative FDR, which in turn results in the desired threshold that serves as a quality threshold for our identifications. Now, I want to present some results by comparisons of our methods. We compared against, in total, against 4 different methods, but here, due to lack of time, I will concentrate only on the comparison against the 6-frame translation coupled to a standard MSGF plus search. And the first dataset I want to present to you is a prokaryotic dataset that is quite well annotated. So a dataset of Bartonella henselae, where we used over 1 million spectra and over 50 million reads, and where we have roughly 1,400 annotated genes. And what you can see here are the result of the comparison. So first of all, MSProGene constructed a database which is a magnitude, an order of which is far smaller than the 6-frame constructed database. And what we can also see is that in total, MSProGene predicted less proteins, and the recall of MSProGene, as you also can see in the ROC curve, is a bit smaller than for the 6-frame, but we identify these less proteins with much higher confidence, so the precision is better than for the 6-frame approach. And in total, or in combination, this results roughly to the same area under the curve. The second thing that that we found for this dataset was that we were able to identify novel genes on the Bartonella henselae strain. Other methods failed to identify these novel genes because first, they were supported by shared peptides, and usually these shared peptides are just excluded from the analysis or result to protein grouping. So we were able to actually count the support of these peptides. And also the chosen FDR cutoff of 1% was too conservative for the other methods. So peptides supporting the novel proteins actually had scores below the threshold. And as you can see here, so we tried to confirm the novel proteins using blast search, and this resulted in quite high identities. So they might be promising candidates. The second dataset is a Lycomuseidis sigmodontis dataset. Here we compared again to the 6-frame translation. This dataset is very poorly annotated, so we have only 75 proteins that are already known so far, so there's no suitable reference database available, so standard methods could not really be applied to this kind of dataset. So here for evaluation, we use the BLAST search to compare the method or the proteins identified by our method and the 6' translation against Brugia malai as the closest relative. And again, you see that the database reconstructed by MSProGene is much, much smaller than for the 6'. And in the end, we identified, again, less proteins, but with much higher confidence. So if you look at the BLAST scores of the resulting proteins, you see that they're a lot of higher than for the 6' identifications. So in the end, looking at high scoring hits, you see that we identify an order of magnitude more hits. And with that, I want to conclude. So I showed you a new method for integrative proteogenomics that works beyond the 6' translation and is independent of existing reference databases. And by that, it identifies also novel proteins or allows the identifications. And it allows to construct customized databases which are smaller and identify at higher precision. And we simultaneously perform peptide identification and protein inference, which also allows to resolve shared peptides and protein and avoid protein grouping. With that, I want to thank my former colleagues from the Robert Koch Institute, and in particular my former supervisor Bernhard Renard, and also people from the Free University of Berlin and my colleagues from Nexus. And I want to thank you for your attention, and now I'm happy to take questions.
I can't see from here, so please, if you have questions, try to wave at me. I do have one in all this, because at one point you said that, if I understood correctly, that if you had two peptides with a frame shift mapping to the same protein, you would discard, you would assume they were wrong. But what about alternative spliced isoforms?
Alternatively spliced isoforms appear as different transcripts in our network. So then it would be transcript 1 and transcript 2. So
even if they're unknown?
Yeah, because the methods, for example, cufflinks, they reconstruct alternative isoforms as separate sequences. So yeah, they are individual sequences. Yes? Yeah. What do you mean, the BLAST score, or? Yeah, we used, to avoid a bias. Actually, if we would use the usual E-value of a BLAST comparison, then we would bias against the 6' translation, because the reconstructed database is much bigger. So that is why we chose the bit score, because the bit score should only, or is not representing or including the database size. So yeah, we accounted for that. I do not have such a plot, but this is an eukaryote. So yeah, the Litomusoides is a eukaryotic organism. Yeah, thank you. The question was, how important is it to have RNA-seq data from the same sample, if I understood. I mean, of course, it's favorable, so it gives you, or should give you, better results if the reference genome, the RNA-seq data, and the MS2 spectra are all derived from the same organism. But, I mean, if you think of it, you could also use related organisms and see what you get. So sometimes it's not possible to, or you have no sequence available, but if you have a relation, or you have a closely related organism, I think you can still try, and you will have, maybe you will have more, or you will have more variants that you observe in your mapping, but it's still, I mean, you can still reconstruct a transcript database based on the mapping. So yeah, you can still use it. But of course, the results will not be as good as in the case of similar data sets.
Okay, I think in sake of, thank you very much again,
Yeah, thanks.
Franziska.